Today we're going to give you an overview of how to use all the equipment that we use for our snapshots. So, Partners of Scott County Watersheds, our goal is to educate the people of Scott County about the importance of water quality. So we do this through a bunch of different ways. First of all, we have monthly educational forums. So at the Eastern Avenue Library on the third Tuesday of the month, we'll have a guest speaker come in from noon to 1, and they will talk about different types of conservation topics. They don't necessarily have to be related to water quality, but we try to bring it sort of around water quality, so soil management, practical farming, practical lawn maintenance, and that sort of thing. Then we also do these 3 snapshots every year. So it's called a snapshot because it's a bunch of volunteers going out at the same time and taking water samples and then coming back and sharing their results at the same time. So all of the data we're getting is from one day in a 4-hour time frame. So we usually have about 35-ish volunteers that will test around 54 sites in the county, and they're all the little tributaries that flow into the Mississippi. And so we have about 20 years of data that are our efforts, plus when the DNR had their Iowa water quality monitoring program that we have a database for. And so what we do with the information that the volunteers collect is we will put that into our database and identify trends in each of those sites to see if there's any problems that are arising. So that way we can figure out if there's a way to fix them. So you guys play a really important role in helping this organization gather this information that otherwise wouldn't be gathered because the DNR doesn't have the funding to do this anymore. And a lot of counties in the area don't have the time or the money to do this either. So with that, I'll go ahead and get started. There's a few different things that we test for water quality. So the first is physical, and that's characteristics of a watershed in the stream channel. So first of all, a watershed is the area of land into which after a major rain event, all of the water will drain into a body of water. So for example, a riverbed, so the valley, that's the watershed. Everything that happens when a rain hits, it's going to flow into the stream. So the physical characteristics are basically you get to your site and you look around, you see, okay, are there any farms near this body of water? Are there a bunch of parking lots near this water? What's the weather like? What else might contribute to pollutants around this body of water? Then we have chemical, which is what you see in front of you right here. We have some different chemical tests that we do. We have pH, nitrate, phosphate, dissolved oxygen, ammonia, chloride, and a few other things. And then we also have biological. And so biological is looking at the little critters that live in the body of water and seeing what the variety is, because if we have a larger variety of animals living in the water, that indicates a healthier stream. So what we do is we take a quick little inventory of the animals that live in the stream, and that's another way we can tell how healthy the body of water is. You guys are probably aware of point and non-point source pollution, but just for the sake of reviewing, point source pollution, we know exactly where that pollution is coming from. So for example, if this was a pipe that was connected to a factory, we know exactly, if we detect pollution, we know exactly where that's coming from. So with non-point solution, that means we're not exactly sure what the source of that is, because after a major rain event, you could be picking up things from nearby farms, parking lots, houses, the street. So you might detect a pollutant, but you might not be able to figure out exactly where that came from. So in the case of the snapshots, that's normally what happens. We detect whether or not levels of chemicals are high or low. We can't necessarily detect where those are coming from, but we can think of ways to maybe bring some of those higher levels down. So nutrients are a big one that we test for. The biggest ones are nitrogen and phosphorus, and those are used not only in lawn fertilizers, but also agriculture. And so they are naturally found, and they help plant growth, which is a good thing. But sometimes you can have too much of a good thing. You guys have probably seen the red tides on TV, and you've seen at Westlake how they had to drain it in order to take care of some of the algae blooms that were happening there. And so that's all a result of excess nutrients flowing into the water. So this is just an example of it's possible to have too much of a good thing. So water quality can be affected in some different ways. For our snapshots, we test 3 different times of year in order to compare what the levels of each parameter is based on the time of year, because it can change. For example, with chloride, which is salts, those are going to read higher in the wintertime. Can anyone guess why? The roads, yep. So that's just an example of how the season of the year can affect what the parameters read. We also have time of day, because as the sun rises, it might heat up some of the water. Also might have trees around that shade parts of the water where you're testing. The weather obviously is going to change, and then any physical influences or land use. So if there's construction going on, that's going to have more soil being moved around that might flow into the water and that sort of thing. All right, so why do we want to monitor water quality? So first of all, we want to establish what our baseline water quality is. The state of Iowa does have certain standards for some of the things that we test for, not everything, but there are different levels that have been approved for human health as well as environmental health. And then we want to identify the trends. Like I said before, we have about 20 years of data that we have collected for Scott County, so we want to see if those levels are staying stable, or if they're rising, or if they're falling, and then locate any issues and possibly mitigate them. Also, we want to build a collective stewardship. Like I said, the effort that we do now is all driven by volunteers. So it's very important to have people like you on our side and willing to take your time and your knowledge out to the field and gather this information for us. It also lets you have a direct hand in helping a problem if one arises. Okay, so here's another example of what a watershed is. Watersheds can be big and small. So this is just an example of, you can see on the left-hand side of the United States, we live near the largest watershed in North America, which is the Mississippi watershed, and you can see at the bottom there by Louisiana, that's what's called the dead zone, and I'll talk a little bit more about that later, but you can kind of see where that is. And then we zoom into Scott County, you can see it's divided into a bunch of different colors, and those are all of the different watersheds that are in Scott County. And then you can zoom in even further, and on the right-hand side, you have Westlake Park, and that's its own watershed as well. And you can even have even smaller watersheds in your backyard if you have a little creek. All right, so how do we measure water quality? We do have a couple different measurements. For temperature is going to be degrees Fahrenheit. We also have centimeters for our turbidity test. But for our other chemical tests, we're going to be figuring out how many milligrams are of a certain parameter are in a liter of water. So when we're showing the different number values on the test bottles, that's what we're testing for, is milligrams per liter. All right, so now I'm going to go ahead and just give you a little bit more information about each parameter that we test, and then we're going to show you how to use the different equipment, and then the last part of the program, you guys will have some hands-on time so you can play with it yourself. So first of all, temperature affects the way that aquatic organisms survive and thrive in the water. They have a very narrow window of comfort, so if it gets too hot, some of the animals might die off. So it also can affect the toxicity of certain pollutants that can affect the health of these animals. And so obviously we want to have as many animals living in the stream as possible, because they provide food for other animals, and they also help break down other things. So like I said before too, cover or lack of cover can affect temperature. So if you're in a stream that is heavily shaded, that will cause the water to cool, but maybe further down the stream, it's out in the open sunlight, so that's going to have a different temperature reading. So when we are deciding where we want to test for water quality with our snapshots, we take that into consideration, and we may even test multiple sites on the same creek. All right, so dissolved oxygen is another important one. Obviously, fish need this to breathe, and so do some of the other organisms that live in the water. And so it's good to have higher dissolved oxygen. A lot of this happens when water is flowing. You can see on the top there, the flowing water will increase the oxygen because it's creating more bubbles as the water flows through. If you have stagnant water, that can decrease the oxygen level, as well as when we talked about too much nutrients in the water. What they can do is they can drop the oxygen levels in the water, and that will affect the health of the plants that live in the water, as well as the animals that depend on them. So here's just an example of how dissolved oxygen can change over the course of a day. So you can see at dawn it's going to be lower, and then as it reaches dusk it's going to reach its peak. So that's another reason that when we're testing, if we see a weird reading, we need to take into consideration what time we tested it and maybe go back and see if it's a different reading during a different time of day. And as you can see, there are Iowa standards for dissolved oxygen. So for warmer water, we want about 5 milligrams per liter, and for cold water, we want about 7 milligrams per liter. All right, so pH level is another important one. A lot of aquatic organisms want to live in this neutral zone of about 6 to 9. Neutral is 7. The pH scale is from 0 to 14. So as you can see on the chart there, 0 is acidic, all the way up to 14, which is more basic. And also this can affect human health, whether a water body is more acidic or more basic. So this can be determined by the surroundings of the watershed. So for example, metal particles that's released in the water can lower the pH, or if you have higher ammonia or other types of nutrients that can change the pH level as well. So chloride, we kind of mentioned this already. It's found in road salt, and so we test for that as well because obviously we live in a freshwater area and we want to keep our waters fresh. So we want to make sure that these streams are not getting too salty because the animals that live in them won't be able to survive. All right, so then we have nitrate and nitrite. Those are both parts, are different types of nitrogen. And those, like I said, are an essential plant nutrient that can cause excess plant growth. So a lot of times people will fertilize their lawns to make their lawns nice and lush, or farmers will apply this to their fields so that their crops can grow bigger, which is great as long as you do it a responsible way. Because if you are putting way too much fertilizer on either one of these, the plants can only soak up so much of that fertilizer before it says, I'm good and I don't need any more. And so when there's a big rain event, whatever's left over is going to get washed away to the nearest body of water. And so that's when it starts to cause problems, because it will cause some more plant growth in the water, and that can affect, kind of do a domino effect, where the oxygen levels are low in the water, which means that the plants that normally live in the water can't survive, and they can't produce more oxygen for the invertebrates and the fish to breathe, which means that we won't have those animals living in the body of water anymore. And then ammonia is another one. It's another, can also be found in fertilizers. It also can be toxic to fish at certain levels. So phosphorus is kind of like nitrogen. It's an essential nutrient. The sources include human, animal, or industrial waste. You can also find it on certain soils and rocks. So at a major rain event, that can also be washed into a body of water. Causes excess plant growth. Here's why it's so important that we keep our phosphorus under control. 1 pound of phosphorus can produce 500 pounds of algae. So if we detect even the slightest indication that phosphorus is growing in a body of water, that's cause for concern, because that can exponentially cause plant growth. So here's the dead zone that I was talking about in the Gulf of Mexico. So we live in a really important part of the country when it comes to watershed health, because we live so close to the Mississippi River. So even though you might think that some of these tiny little streams and tributaries that are in Scott County don't matter, they really do. Because if we aren't paying attention to the pollution levels that are in these little streams, they're eventually going to run into the Mississippi River. And then after that, whatever runs down through the Mississippi River and collects along the way is going to go into the Gulf of Mexico. And there is a large area of water in the Gulf of Mexico in which they call the dead zone, because there are no plants and animals that can thrive there. And that is a major result of excess nutrients in the water. So it's basically collected over time as it's gone downstream, and there's a big bloom of algae over the water that blocks out the sun, and like I said, that can create a cascading domino effect, and there's no animals that can live there, and it continues to get larger. So it's really important for everyone upstream to be conscious of that, because if you don't do anything about it, it's just going to get bigger. So another thing we test for is transparency or turbidity. So that's basically how clear or how murky the water is. So the way we do this is we have a tube that I'll show you later, and it kind of gives you an idea of how many sediment particles are floating around in the water, or how dirty it is, basically. So sometimes you can see when there is a high rain event, you can see a raging creek, and you can see that it's pretty dirty because it's picking up a lot of dirt along the way and causing that dirt to be pushed downstream. During calmer waters, or if the stream banks are in really good shape, that water should be clearer and not taking a lot of particles with it downstream. We want to try to keep the soil in place where it is, especially when it comes to agriculture, because the topsoil is where all the nutrients are, and that's what the farmers use to make good crops. If there's a lot of erosion happening, that topsoil is going to get carried away downstream, and then as a result of that, the farmers need to put more fertilizer on their fields, and then it causes the cycle of the nutrient loss with the rain event again. So it's really important that we keep track of what kind of erosion is going on everywhere so that we can keep that soil in place. Also, turbidity and transparency can indicate where there might be possible flooding or redirecting of stream flows, and that can cause problems that we all are aware of when sediment will settle somewhere that it causes the water to be rediverted somewhere else. Then finally, we have biological. So that is calculating what kind of critters live in the water. So you may not be aware that there are a lot of different animals that live in our streams. And when you get a chance later, you can look at the chart that we have up here at the front. So this is a few drawings of the animals that live in our creeks, and this is not to scale. They're all a lot smaller than this, although some of them are a little bit bigger than this. But they're all really important in the health of the watershed because they rely on each other for food. They can break down different things that are in the water for us. And they also provide food for fish and other organisms. So they're a very important part of the ecosystem. So what we do is we will take a sample of water, and then we will pick through the water and see what we can find. And we have a measurement. It's called a biotic integrity index, and that kind of scales what the diversity of that particular watershed is. Okay, so what's in your kit? So every group of volunteers that goes out and tests for the water will get a mesh bag that is full of testing equipment. And so that's everything that you need in order to do the water quality tests and then mark your results down for us. So first of all, this is what the bag looks like. We're probably going to get some new ones hopefully in the future, but you get a mesh bag with all of your things in it. You'll get a bucket like this, because with the case of most of the samples that you're going to take, we don't want you mucking around and jumping in the creek yourself. Not only is it dangerous during certain times of the year, but it's just a lot faster to use a bucket instead of jumping down there yourself. So this bucket will be attached to a long rope, and most of our testing locations are near overpass, so with a curb. So you can get out and get on the shoulder. You've tossed this bucket over into the creek. This little carabiner will weight it so that it can collect water. And then you only need maybe about half full of this. You pull it back up, and that's where you're going to take all of your samples from. So that's a lot easier way to do it. This is your turbidity tube. This is how you're going to tell how clear or how murky the water is. And there's a symbol on the bottom of this tube. And that's how we're going to measure when we stop the water flow here. So if I have a really clear body of water and I look in here, I should be able to see that symbol right away without letting out any water. However, if the water's super murky, I have a buddy, or I can do it myself. You release some of the water out of this tube until you can clearly see that sign on the tube. Then we take a measurement here of how many centimeters it took for you to see that sign. And that's how you measure turbidity. So that's a really easy one. All right. We also have a waste container. So anytime you're dipping some of the test strips, you have your waste container. You have your different chemical tests here: pH, nitrate, chloride. You'll probably have ammonia too. You also have these 2 boxes that have your test kit for dissolved oxygen and phosphorus. You'll also have a clipboard with a pen and maps and driving directions to all your different sites. And then you'll have your worksheets that you're going to write all of your results down on. If you're doing biological, so I forgot to mention this earlier, usually on our Snapshot days, you'll participate in either the chemical or the biological because there's just not enough time to do it all. So if you are having a biological team out there, you have this little basin. So what you'll do is you'll collect, this is where you actually do some creek stomping. You'll collect a sample of water, make sure it's nice and clear and there's no muck in there. Then you can take one of your large nets that you're provided with, and you'll scoop material into there, and you will sort through it with your hands if you're brave, or we can get some tweezers for you too, and you're going to identify the bugs based on the chart that is provided for you. And then we also have a tarp to clean some stuff up with, some baby wipes for you, and then we have ice cube trays because that's a great way to just put individual animals in and separate them so you can identify what they are. So that's what's in your bug kit. All right, so in order to test for dissolved oxygen, we have this cool little box, and it has everything you need in there. So first of all, you have a sample tube, and you'll take the sample from the bucket that you got originally, and you fill this up to 25 milliliters. The directions are all in these kits too, by the way, and we'll also have them written out on your clipboards. So there'll be a variety of different ways where you can find directions on how to use these. But you fill this 25 milliliters. Then you have little pipettes, or not pipettes, what are these called? Ampules, yes. Ampules in the box. And they're glass, so you have to be very careful when you handle them. There's an activating solution in here. You will dip the ampule into this little vial, and there are actually little divots on the inside of this vial. I don't know if you can see that or not, but that's where you can put the tip of this, and then what you do is you put it against the side and then push it until the tip of this ampule breaks off. And what will happen is that will force water up into the ampule, and it will mix with the activating solution and turn a shade of blue. Once it turns a shade of blue, I think you do this for about 2 minutes, kind of like a blood sample. You mix it around, get that little bubble to flow back and forth. And then after exactly 2 minutes, timing is very important with some of these tests to make sure that you're getting an accurate reading. You set it against this little comparator chart and see what color blue it turns. And obviously, it's just your best guess. We are relying on you as a volunteer. You don't have to be an expert. Just give it your best guess for us. And then you write down your results. So that one's super easy. We've also got pH. We test pH with these little strips. What you do, you have this little strip with a little foam pad on the end of it. So you will dip this in your water. It says here for 15, or dip it, move it immediately. Hold it level for 15 seconds so that the pad is face up. After you count to 15, what you'll do is you will compare what color it changes to the bottle. And once again, your best guess. And then you'll write down your answer for us. And then any waste you use goes in your waste jar. We don't want to litter while we're out doing our water tests. All right, so chloride is a little bit different. This is one that we recommend for volunteers to start off with because this test takes the longest out of all of them. So this one also has test strips. What you do with this one is you can get another little beaker that's in your kit, or if you have an extra one of these, you can fill it up with water. You don't even have to fill it all the way up. You can just kind of put about an inch worth of water in there. On this strip, there's a little yellow stripe on the top, And you will place the strip in there and just let it wait until that strip turns black. Once it turns black, then you know that it's ready to read. And what will happen is you can kind of see on this TV screen, there'll be a white kind of arrow that will appear on the bottom of the strip. And that is where you're going to take your reading, at the tip of the arrow. So there's a little bit of conversion that's involved, but it's all on the bottle. So once again, no math required. So for example, if we have a level of 2.9 on here, we would look at the bottle and convert it into milligrams per liter. So this goes by even numbers, so we'll say it's 3. So on the bottle, it says it's 473 milligrams per liter. So the bottle will tell you how to convert it for our records. Okay, so nitrogen is another one we use the strips for. It's a very similar way with pH, except that you have 2 colors you're going to compare this time. So that means there are 2 strips on the, or 2 pads on the strip. So you dip this in the water for 1 second, bring it out, hold it level like this for 30 seconds. Once again, timing is very important, so you want to make sure that you are reading this right after 30 seconds. Then you're going to hold it like level like this so that you can compare the 2 different lines. Now, you're not guaranteed to say, okay, well, I have a 1 on the top. That means the bottom has to be 0.15. They're not necessarily going to match up. So you can first try matching the top one and then matching the bottom. Okay, ammonia, I don't have one up here with me, but it's the similar sort of thing where you dip it in the water, you have to hold it level. This one, it says to vigorously move the strip up and down when you're testing it. And then once the time is done, then you just take a reading on the bottle again. And then phosphate is similar to dissolved oxygen. There's just one extra step that's involved. So you have another little kit like this. And you would take a sample, about 25 milliliters again in your little jar. Before you put your ampule in there, you have an activating solution. And so it's another activating solution that the dissolved oxygen doesn't have. So it's kind of like an eyedropper, but it's really thick, so you kind of had to squeeze hard. You put a couple drops of that in the water. And then, just like dissolved oxygen, you have these ampules that you will put in here. You'll put against one of those little divots in here again and break off the tip. Once again, you're going to move it up and down like this to get it thoroughly mixed for a couple minutes. Then there are a couple different ways that you can read phosphate. So, oops, there was another one in there. We recommend using this one first. This is smaller increments to measure than the one that's on the lid. So what you would do is you would put the end of this like this. Make sure you're looking out of the end that is not open. And you will compare what color blue it is. There's another wheel of different colors in here. And if you hold it up to kind of a neutral light source, that will help you determine what the value is. If you can't get a good reading off of that, then you can also use this, just like with dissolved oxygen. All right, so transparency, I kind of already went through that for us, but once again, pour the sample in here as far as you think you're going to need it. If you think it's really clear, go ahead and fill it all the way up just to be safe. If it's really murky, you could probably start it like somewhere in the middle. And then you're going to release the water until you see that symbol at the bottom of the tube. And then biological, you can see the pictures here again. This is from last year's Snapshot in July. So they took a sample in the nets. They broke it apart inside of the bucket. As you can see, sometimes you got to get a little bit dirty, but it's fun. And you can find a lot of animals that you weren't aware of that lived in our area. And then you can compare it to the chart that we have here. So that's how that works. And that one does require a little bit of math in determining the biotic integrity index. But as far as the biological testing, we always try to have an expert, or at least someone who has done the biological testing before, to help you out with this, because as you can see, sometimes it gets a little tricky to identify the difference between these. All right, so just kind of as a summary, you can see water quality is detected by a lot of different things. We have our physical parameters, so what's going on outside of the watershed or around the watershed. We have our pollutants, toxic and non-toxic, as far as habitat. So what's going on as far as where the animals are living in the water and what kind of animals you see in the water, and then a couple of other different things. So here's an example of the data sheet that you fill out when you are going around to your different snapshot areas. So I also have them printed off at the table over here that you guys can look at later. So we have about 54 different sites, and they're all indicated by a 6-digit number that we've determined that was preset, I think, by the DNR when they were doing the Iowa water testing. And so that's the first thing you write on your worksheet. That's the most important thing you write on your worksheet, because we want to make sure that these numbers are getting matched to the correct site. Then we have your guys' names, when you took it. Like I said, water quality can be dynamic as far as time of day and time of year. And then this is the physical part. So at the top, you're going to look at what the weather's like, what the temperature of the air is, if it rained in the last 24 hours or 48 hours, what the water looks like, if it's murky, if it has a weird smell, if there are animals in it or near it, what kind of land use is around the body of water. And then you can get into your chemical tests. And then when you write your values down, we already have the values listed for you, and you just check the box for what you find. And then here's an example of the biological introductory assessment. So we have all the different bugs listed, and then you count how many you found of each bug. And then once again on the top, you're going to write your site number and the time of day, and then there's a little bit of math involved with determining the biotic integrity index. So this is something that I'll review with all of our volunteers on the day of the snapshots, but it's also something that you can keep in mind here. So I have some best practices that I've kind of come across as far as the last year that I've been doing this as my coordinator. First of all, we do have a waiver that you sign, so you have to sign that first. I provide driving directions and maps for all the groups so that you don't have to rely on technology necessarily to find these sites, because some of them are pretty rural and you may not have great cell phone service. So I try to give you turn-by-turn directions, as well as if it's not like right on the road and you have to park here and walk a little bit, I try to give you directions for that too. So each site is designated with a stake that has 2 yellow ribbons on it. So a few days before the snapshot, we go around and we put stakes next to all of our testing sites, so that as you're looking around and you see the stake, you say, okay, that's where we should park and that's where we should test. I try to put the stake on the side of the road that I want you to take the sample from. And also, I really appreciate it when the volunteers collect the stakes as they go, because then I don't have to drive around and find them. And then we do test a couple lakes and ponds. I don't stake those because it's pretty obvious when you show up to a lake or a pond. Some of the sites are in areas where parking might be difficult or dangerous, especially in some of the rural areas because there isn't a shoulder. So just take that into consideration when you're testing. You may want to park a little ways in a safer spot and walk to your site, or just park as close to the side of the road as you can. Maybe have a couple of your volunteers kind of as lookouts for traffic so that you're all paying attention to what's going on. We will have safety vests for you to wear as well. But in any case, if you don't feel comfortable where you are testing, it's not life or death for us to not have data for that site, so we would rather have you be safe than nervous about where you're testing. And then carefully read the instructions for each test. Like I said, we'll have a quick overview before you go out to your snapshot. We'll also have all the instructions for the different tests written on a sheet in your clipboard, and you have each direction for the tests on the bottles and the cases themselves. Depending on the time of year, you might run into some noxious weeds or bugs. So for those of you that aren't familiar with what poison ivy looks like, leaves of 3, leave it be. Basically, the 3 leaves all have little mittens. They kind of look like hands. So you have your 2 here, and then the middle one will have 2 little thumbs in the mitten. Wild parsnip, bad news. It is green, and then it has yellow kind of flat on the top. It almost looks like a Queen Anne's lace, but a little beefier. You definitely don't want to get in touch with those. If you are exposed to sunlight and the oils to those mixed, it can cause some really bad burns. So just be aware of those. Once again, as far as safety, if you come across a site that you're going to test and you see all of this stuff everywhere, don't even worry about testing the water. Just skip that one because we don't want you to get injured. And then same with ticks. Just give yourself a tick check after you're done volunteering because they might be there. All right, so I'm going to say this again. Always, always, always write the 6-digit number of your site on the worksheet before you get started with any tests. This is the number that we need the most because that way we can attach the data that we're getting to the site. If we don't have this number, then the data that you collected is null and void because we don't know where to put it. And so make sure it's on there. And also write your results clearly so that we can read them. We occasionally will have some of Amy's interns input the data, and so it's a different person reading it every time, so make sure it's nice and clear. Depending on how many people are in your group and how comfortable you get as you're testing these sites, you might want to divide and conquer as far as doing the tests so that you aren't spending a whole lot of time at each site. So for example, if you have a team member that really likes doing dissolved oxygen, you just say, okay, you'll do that one at every single one. And while you're doing that one, I'll go ahead and test for chloride. And then maybe while I'm doing that, someone else is prepping for phosphorus so that you guys can kind of get it done in a more quick manner so that you're not doing one after the other. I always have my phone number on all the clipboards so that if you are lost or not sure where the stake is, I can help you out. And then these snapshots always run from 8 to noon, and we want you guys to be back by noon regardless if you're done or not. Like I said, you can try to be efficient and do more than one test at a time, but if you find yourself running short on time, we just want you to come back because we provide pizza to all of our volunteers as well as thank them, and we don't want you to be out there all day. So we want to make sure that you get the recognition and the pizza that you deserve. So once we collect all of this data, we will share it with you guys so you can see the results of your hard work. And we will also put it on our website. We recently got these new cool maps that I'll talk about in a couple of minutes that show all of the data that has been collected. And then we will use this data for further grant applications so that if we have a situation where we think that we can help maybe lower the levels of pollutants in a certain area, we can apply for grants to fund that project. So that's where a lot of this data gets put to use. So here's what our maps look like. If you go to partnersofscottcountywatersheds.org, you can find these maps, and they're interactive and they're really cool. They were just debuted a few weeks ago. And we have 2 different types of maps. So we have kind of an introductory map, and then we have an in-depth map. So the introductory map is the one you'll see right here. And it has all of the watersheds in Scott County and a little bit beyond designated by dotted lines. When you hover your mouse over them, it will highlight, and then you can click on it and it will tell you which watershed that is. And then on the side, it will give you some stats about how many acres that watershed is or square miles, what percentage of that land is used for ag, for industry, for residential, that sort of thing, as well as what streams are in that watershed and where it drains into. The second map gives you our snapshot data, so you can see all those little green dots on the map. Those are all of the spots that we normally test for our snapshots, and you can get really, really deep into the information using this map if you want to. So you can click on individual sites and you can see all the data that we've ever collected for that site in a couple different charts. Also, there's some visuals that kind of give you trends. There's line graphs that will show you trends over time for certain parameters. And you can even filter it by certain dates that you want to see the results from, or even certain streams, so you can filter out some of those, or even a watershed as a whole. So even we are not experts on this yet. There's a lot going on. We have the Scott County GIS guys to thank for creating this for us. And it's a great way for us to visualize the data in a way that's more user-friendly than the spreadsheet that we had before. That was a little harder to read. So here's an example of ways that we can use the data collected to fund a project. So we've noticed through our data that Robin Creek in Northwest Davenport, kind of behind Popeyes, was starting to have higher levels of fecal bacteria indicated. Not only do we do our normal snapshot tests, but depending on funding, we can do different types of lab tests for herbicides, pesticides, bacteria, and some other things that the staff will do. The volunteers don't do that part. But anyway, we found higher levels of fecal bacteria in Robin Creek, and we were trying to figure out if there was a way that we can reduce those. So Steve was able to find this kind of revolutionary project that is going on in Ohio, in which they made these things called bioremediation filters. And if any of you have visited Middle Park Lagoon or Eastern Avenue Park, or even out here at Nahant, we have these artificial islands that are floating, and they help reduce some of the bacteria levels and the algal blooms that are in the water. So it's made out of the same material as those floating islands, but these bioremediation filters are inserted into the stream so that the water flows through them rather than having the material sit on top of the surface of the water. So the way that it works is you divide this material. It's kind of like a steel wool, but it's made out of plastic, and you chop it up into strips. You can insert it into the stream. Once it's in there, plant material will start to grow within this matrix, and that plant material can help break down the bacteria as it flows through the stream. We're going to insert these filters in 3 places in Robin Creek. And hopefully as the water flows through each of these 3 filters, it will reduce the bacteria levels. So we were able to take that data, apply for a grant with Iowa American Water, and they, along with the City of Davenport, are going to fund this project. And so in the springtime, we're going to first test the water without the filters. And then once we insert the filters, we're going to do regular testing so we can see if they're doing what we want them to do.